and tutorials left, but the exams we conducted for 4 days and it's over fortunately yesterday, and I also gave you the date sheet, so now it's the time to give you the certificate for the course. So this course is also recognized by MHRD, and you can also claim a credit. Whoever is attending, you can actually upload the certificate and your grade certificate that you get from your college or university, you can claim a grade credit from this.
MHRD does not exist anymore. MHRD does not exist. Ministry of Education.
Ministry of Education. Thank you. Ministry of Education. Thank you. So now I will just call the name, and I request sir to first say a few words, and then we distribute the certificate.
So, good morning to all of you. At the outset, my apologies for not being there on the first day, because I should not say that I had a more pressing responsibility, but all the heads of the institutions were invited at the Rashtrapati Bhavan, the President of India's house, and there was a conference there, so I was supposed to attend that. This was on 3rd and 4th of this month, in the beginning. I greatly admire the efforts of Tripta. I know how difficult it has been, and to organize even the small meeting like this at a small scale, a few participants, and the endeavor of the institute is to make the things more simple, more easy to do, and hopefully other faculty who will plan to organize such things will have easier times, because we are doing certain things for the future. Welcome to the visitor from Bochum, Germany, and you have taken time to visit us. That's great, in the sense that in the present day world, which is leading towards division, these kind of visits bring unification, and they connect people of different countries, and the knowledge as well. The kind of expertise which he brings from Germany is ready to share it with all of us, and therefore we are grateful to you. The topic that lipid membranes, what is that?
From cells to synthetic biology.
Yeah, from cells to synthetic biology. Okay. So this is truly interdisciplinary in the sense. So biology and physics, and a lot of great things come out when people from different disciplines, they come together, start talking to each other, and understand each other's language. Because I do not know much about it, but what I know a little bit is that solving the structure of integral membrane proteins has been a great challenge for many, many years. And developing crystals and then solving this structure, that was a very, very challenging thing. How do proteins integrate into the membranes? How do they fold in the membrane? Different principles apply. Why the membranes are on the outside, although in the eukaryotic cells they are inside as well, but in most bacteria you find them, prokaryotes, they are on the outside, not inside. And what features of the membrane, particularly the outer membrane, makes it useful for being on the outside, and what features make it oriented towards inside. These are some of the questions which I sometimes think about it, and the membrane vesicles which are being used to deliver drugs in different parts of the different tissues, different parts of the body. They have been of great use. I'm just trying to recollect some of the things which I know about the subject a bit. So these are some of the challenging things, and I'm sure that the talk by the experts from Germany and from our own institution will enrich your knowledge to take lead and answer more challenging questions, more original questions. And I'm sure that this training might have helped you to think originally. Not the repetitive research, what is being done already by some people, you continue to work on that. Instead of that, thinking some new original questions and trying to answer them. So with this, I congratulate all of you who have attended this program, and wish you all the best, that you do great in the future. Thank you.
Thank you, sir.
Thank you so much, Professor. It was really a great pleasure also from our side, and we really appreciate also the support by the Institute. And I think I have to thank all of you, because it was fun to teach, and also for my colleague Denise, it's always fun to teach. And we were very pleased. The longer the course was, the more interactions we got. Yeah? Some people already in the very beginning were very open and contacted, and others warmed up during the course. We learned also some things from you, so don't worry, it's for us always an enlightenment to talk to basically people of physics, mathematics. You have an extreme knowledge in physics and mathematics, which I hope you could see will be extremely important to solve the next questions that are all waiting for us. And I hope I have inspired you not to be in fear of working together with biochemists or with chemists. If I have achieved this with my colleague Denise and also with Tripta together, then I think this was actually what we wanted to achieve, that you basically break over the borders and look also into chemistry. Maybe you remember now also a little bit more the biology that you basically might need in your future approaches. So thanks for that, and of course we are not yet done. After this session we will continue a little bit to have fun. So you can enjoy, and I will still a little bit work after the ceremony now. Tripta, also to you, special thanks.
So you have to be around for the certificate distribution. For sure. Yeah, you are the one who transfer the knowledge, your experience to them.
So now I will just call some names. Please come and collect the certificate. Thank you, Thomas, for your words. So Rajni from IISER Mohali. Mahesha from Central University of Karnataka. Naman Srivastava from MIT University. Rupi Bharti from Ahmedabad, Gujarat. Chetna from Bangalore University and IISER Mohali. Sugata Chaudhary from IISER Mohali. Can somebody take for him? No? Okay, I'll go. Charu Taneja from IISER Mohali. Suchita Mishra from IISER Mohali. Devansh Kaushik from Shiv Nadar University. So also Bashir from Shiv Nadar University. So Hindol Chakravarti from Central University of Gujarat. Professor Sarbari Bhattacharya from Bangalore University. Arun Kumar from IIT BHU. So Mohit Anand from IIT BHU. Richa Pal from IIT Ropar. She is not here. Okay. Dimpy Devi from IISER Mohali. Diganta Bhaskar from IISER Mohali. Harshmeet Kaur from IISER Mohali. So Tanmay Pandey from IISER Mohali. Sugata Chaudhary from IISER Mohali. Sugata, sorry. Now there were 3 lab sessions. So this is just a certificate of appreciation for student who volunteer for the lab session. So Harshmeet Kaur. Chetna from Bangalore University and IISER Mohali. So Rajni from IISER Mohali. Now Avtar Singh, the staff, Department of Physical Science, IISER Mohali. I also would like to thank Dr. Dennis, who is here from Germany. Can you please come here? Sure. This is a small token of appreciation. So this is a small thank you to Thomas for So thank you everyone, thank you sir, and there are actually many more people who contributed. If I missed anyone, Ram Shankar ji, you have also, we also have to thank him for managing the lecture hall and everything, and also thank you also for recording and everything. So there is a tea break in 5 minutes. Please enjoy the tea and come back here by 10:45 for the next lecture. Okay, thank you.
5 minutes,
15 minutes. Yeah, and also there is a lunch we have organized outside, so all of you are invited. Please do come, and I mean use this time. I hope to discuss with Thomas and also with Professor Tripathi, who is also a biologist. So kindly please use your time and so that it's efficient for you if it helps you later on. Thank you for your time, sir. Thank you. Thank you. So you can take your seat and then, yeah, one last photo. Yeah, of course, we have to have a group photo actually. Please come for a group photo.
Yes, we can try. That's good.
Can we go out or should we do it here? What is better? Outside? Bahar? Bahar thoda chaliye. We will have a group photo.
कपड़े छोड़ोगे
बेटा पीछे टेबल में टिश्यू देगा, टिश्यू होगा सबसे लास्ट पीस पे, कॉर्नर पे नीचे होगा, उसके कीबोर्ड पे रखा होगा टिश्यू। दो पैकेट ले आओ। आज टिश्यू लेके आए मेरे पास होगा।
Is it 45?
Sorry? Yes. Thank you so much for all the help.
I also stuck here for a certificate. I resigned from my internship, so I needed a certificate from back then. No one was agreeing. Sir agreed.
Okay.
So I'm stuck here for 7 days.
Oh.
Today is the last day.
कौन है कौन नहीं है बंद कर दो
हम पीपीटी खोल दो कोई
पीपीटी हाँ
मैं एडिट कर पीपीटी खोल दो कोई भी
नहीं हाँ हाँ तो आप ही प्रोजेक्ट करोगे ना आप नहीं कर सकते ना
नहीं मैं कर रहा हूँ वो क्रॉप करना था उसका लेआउट तो बोला तो कोई भी सैंपल खोल दो एक बार
अच्छा अच्छा
हाँ।
फिर
नहीं हो पान पीपीटी बता दो भाई। हाँ जी सर। देखिए हमारे पास ना कल तो मैंने वहाँ कट के एक स्लाइड ना, तो जैसे ये हमारे पास 4 स्लाइड है। हेलो हेलो, माइक टेस्ट। हेलो हेलो। अभी 4 इनपुट्स हैं हमारे पास। ये वॉइस के लिए है, ये ऑडियो माइक्रोफोन है।
हेलो, हाँ जी सर। मैंने सर उठा दिया सर, जेब में था, जेब से निकाला, वाइब्रेटर रखा था। हाँ सर, मैं देख रहा हूँ सर, उन्हीं के पास हूँ। जेब से फ़ोन भारी से निकाल रहे हैं, पहले के पहली घड़ी में कौन उठाया फ़ोन।
हेलो 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 हेलो।
सर बोल
अब
रहे थे कि
देखिए,
तुमने
अब
कहा था स्लाइड की, तुमने कहा कि ये स्लाइड है, देख रहे हो क्या? मैंने कहा देख तो रहा हूँ बट
4 इनपुट है। ये कैमरा से आ रही है,
हाँ।
ये पावर पॉइंट से आ रही है, जो वहाँ पे डायरेक्ट हमारा कंप्यूटर के साथ लगा हुआ है, जो उन्होंने दिखाना है।
मतलब जो वहाँ से वो हमारे कंप्यूटर पे
हाँ।
जगह लगी हुई है।
हाँ,
ठीक है।
और ये हमने उसको एडिट करके बनाया। एक के ऊपर लगा हुआ है वो कैमरा जहाँ पे बंदा पढ़ा रहा है, दूसरा पे जो वो पढ़ा रहा है। ठीक है, अब इसको एडिट करा हुआ, ऊपर वो नाम लिखा हुआ है, इसको टाइटल वो सर करके देंगे तेरे को।
अच्छा, ठीक है। और
साउंड हमारा इधर से आ रहा है, ऑडियो ऑन है।
Okay, so just to make sure from the time, should I take an hour? Yeah.
Stop.
So, theoretically, we should take 2 hours, but which means we will be late for the bus. So, I think we should try to wrap up 2 lectures, but in between you take a break so that you have that it is the next lecture. And then, so I think the lecture has to be 1 hour, 12 o'clock, and then after that maybe 12:30 we wrap up.
Let's do it like that. So what we will do is basically we go 1 lecture more, and after that we have a tutorial, and we can always stop at any time so that we have enough time also to move to the bus. Yeah?
12:30 we should.
Okay. Okay, so welcome back to the next session. I promised you that today we would basically maybe have few examples how nanodiscs can be used. Remember, these nanodiscs were little membrane sheets that were stabilized by the help of proteins, so-called belt proteins, or by the help of particular polymers. And the secret was that these belt proteins or the polymers, they would basically shield the hydrophobic part of the lipids from the aqueous environment, and in this way would basically keep these membranes in a soluble state, and these membranes could have been equipped, of course, with membrane proteins, depending how you have prepared them. Okay, so what can you do? So let's go back to a membrane, and this is what you see here, our typical biological membrane. It's a layer of 2 lipid sites that are basically here in this case composed of different type of lipids, and imagine you have a membrane protein. In this in particular case, we can take an example of an active transporter. So that would be a so-called ATP-driven transporter that, for instance, moves protons. Where would you find such a membrane protein? You would find it, for instance, in all fungi. So fungi use these type of membrane transporters to energize their membranes, to make an ion gradient, in this case based on protons. In animal cells, this gradient would be based on potassium and sodium. But these pumps you can also find on plants. So they are basically in the plasma membrane of plants, so plants also use a proton gradient. And that's something very important, because we talked about that such a proton gradient can be used to take up nutrients. Such transporters you find, for instance, in the plasma membrane of the root cells in the plant. So what is the task of the root in plants? The root, basically the root cells, they need to take up nutrients. They need to take up ions from the environment. And also these root cells, they need to duplicate, they need to basically drive that the whole branch of roots, it's basically growing. That's a very difficult task, because in the ground, you need to dig into the ground. And you know how difficult it is actually to dig into the ground. Plants do this with their roots. And to do so, they need to develop quite some forces. They need basically to expand these cells. They need to grow. And in this process, these proton pumps are very important. And also, for instance, pathogens take advantage. They either have substance to activate these pumps or to deactivate these pumps, and by that can basically easier penetrate into plants, can better attack them. So that means obviously these pumps needs to be controlled for the actually growth of the plant, and also in fungi these pumps needs to be controlled that the fungi can grow. And for the control we already discussed, there can be several things. These pumps can, for instance, interact with other proteins. We learned, for instance, that could be cytosolic proteins that can bind. And typically these pumps are constructed in the way that they can have 2 different modes, an active mode and an inactive mode. And this inactive mode can be controlled by the pump itself, because the pump can have, for instance, large cytosolic parts that are normally clamped, or they can open. And if such a protein interacts, for instance, with these sides of the protein that are now working like a lid and opens up the lid, the pump can now bind ATP and can start to work. So these would be interacting proteins that can activate a pump, or other type of proteins can inactivate. So those we would for instance, maybe are interested to find those proteins or to understand how do they interact. We also talked about receptors. Remember, this would be for example, a single-spending transmembrane receptor. What would be the prerequisite that this receptor would be activated if it's only a single-spending transmembrane domain receptor? You remember? This one would be difficult with signaling, would have difficulties. It needs two of them. It needs, for instance, either the same copy again to dimerize into a homodimer, no? Because a single transmembrane protein cannot signal alone. It's not basically from the cell. It's difficult to see if a ligand binds on that side. Do we have it? Yes. That actually, indeed, there's a change basically here on that side because it cannot be transmitted unless we transmit it into a way of forming a dimer or higher oligomers. So it's a very simplified drawing, which is typically nevertheless used, but you know already these drawings are only as a type of indication. They not necessarily show the full details of the molecular process that's going on. So be always also critical with drawings. They are helpful, but they are limited. No? It's like also sometimes when you have a drawing in 2D, but in fact you want to draw something in 3D. Also there's this limitation of basically seeing the full object, right? Okay, so now, for instance, we could be interested if those two proteins, they might need to interact. The receptor gets activated and forms a complex with our pump, and in this way would activate the pump. No? What would be the structure of this complex? Because if we would say now, Tanmay would be our transporter, and I would be sitting in the membrane, and I would be the receptor sitting in the membrane, the question is, do I bind in this way to him? Or do I bind in this way to him? Why could this be interesting? Why would Thomas be interested to, I mean, is it not enough to know, okay, we interact? Why would it be interested to know whether I do interact like that or do I interact like that? What do you think? For what could it be helpful to know exactly how the proteins interact on which side? Do you have ideas? Sometimes with life it's always difficult to find some analogies in life, no? But at lunch, maybe you observe the people at lunch. I can give you the information, we were all at the lunch table, right? Okay, then you think we have interacted, right? In a way we have interacted because we have been in the lunchroom. But what if I would have a photo exactly showing where you have been sitting and with whom you have talked? What would this tell me more? The communication. And if you then want to interfere with this communication maybe later, if maybe, Tanmay don't want that we interact.
The forces of interactions.
Yeah, so Tanmay could find a way of sitting between us. So let's say it's an analogy of, for instance, finding an inhibitor. It's a different, basically, when you have this type of interaction, you need a different inhibitor than you would have this interaction, because this inhibitor should be specific and only break this interaction or this interaction. So you would need exactly to know how these interact. It's not enough to know that they interact, but how exactly do they interact. So typically, what we do in science is we go always then one level down, more down to the molecular details. No? In the extreme case, you might end up with quantum physics, but in biochemistry, this is not necessarily in all cases. But it can be necessary that you even go to this level down to understand why is there an interaction, what brings these molecules and holds these molecules together. Okay, so maybe this is a motivation now for you. Okay, how can we study this interaction? No? Can we somehow get insight also in the structure of this interaction? So if we would basically now just use detergents and completely destroy everything and would basically analyze it in a solution, that will not really help because the interaction might be very much compromised, very different. At minimum, you would need to take the detergent away and hope that you get a membrane formed and hope that then you reconstitute this interaction again. No? That would be the minimum. So for instance here, this receptor could be a kinase, a receptor kinase. Remember, they are typically dimeric, or they need to dimerize. And then they could phosphorylate this part of the protein. If this part here gets phosphorylated, it might change the conformation. It might move away like that, and this site becomes accessible, for instance, for binding ATP, and the transporter can start. So what we can do is basically, we take our membrane protein from the membrane, we take our receptors from the basically biological membrane, we take lipids again, mix it, and we start this reconstitution process. And we take our membrane scaffold protein. In this case, we are generating nanodiscs. And you remember, this will happen when we mix this in the right ratio. You will basically, after removing the lipids, get these self-organized particles that we call nanodiscs that you can separate. Just for curiosity, let's say, if you add a lot of lipids, if you add an excess of lipids, plus protein, plus membrane scaffold, and you remove the detergent, what would be the consequence? What do you think you would generate? Just that I get a little bit of feeling that you understand this principle. No? We could say we mix scaffold protein, we mix our purified proteins, and we mix with lipids and detergent, but we take a lot of lipids. And now we will remove the detergent. Who would interact with whom? No? Because when we remove detergent has prevented the interaction so far between lipids, membrane protein, and the scaffold protein. Now we take the detergent out. What will happen? Just come with ideas. It's not so. That could be a consequence. You would basically have a lot of lipids, so they can form maybe a lot of empty nanodiscs. If you run out of membrane protein, if you run out of scaffold protein, but you still have lipids left, and you remove, suddenly you got vesicles. So it becomes very, very important, the ratio now, because if you overdo it, for instance, with the lipid part, the self-assembly will not only generate nanodiscs, it will also generate vesicles. But when will it only generate vesicles? When you have the right combinations of lipids, for instance, cylinder lipids, or you have inverted cone-shaped lipids and cone-shaped lipids that you mix in the right ratio. Otherwise, you might even get micelles or inverted micelles. Remember? So you take advantage of these self-assembly properties, but they not necessarily lead automatically to vesicles or automatically to nanodiscs or automatically to nanodiscs with proteins. Depending on the ratio, we will always get a mixture. That means you always need, in the end, probably to purify the structure that you are interested in. Today, we would be interested in this type of nanodisc containing the protein. Okay, and here you see just an example. We did this for basically a proton pump from the plants. So we separated this. We have here the empty nanodisc, here our nanodiscs that contain the proton pump. And you can follow this by a method basically that is called protein gel electrophoresis, SDS gel electrophoresis, where you can make these proteins, separate the proteins according to their size, and you can make them visible. And you have here the belt proteins, your protein of interest. That's the protein of interest, that's your belt protein. So your protein of interest comes with the belt proteins. And since this is an active transporter, it would hydrolyze ATP. So you also see that basically in these fractions you have a high activity of ATP hydrolysis. So you have now a particle that is formed of lipids, your membrane protein, and your scaffold protein. And the very simple question that we asked here is this pump a single pump in the membrane, or would it form oligomers? Because often a phenomenon in biology is that you have a membrane transporter, copy 1, copy 2, copy 3. You are active, I'm active, you are active, but we are sitting together. And we don't know whether to be active, the prerequisite is to form this oligomeric structure, or whether the single protein would already be active. For the proton pump, it was found when you take membranes and you make photos, it's actually organized at hexamers. 6 copies are sitting together. Is this the prerequisite that the pump would be active, or would be the monomer already be active? Now with this system you would have basically the monomeric protein in the nanodist, because we selected such a small belt with so few lipids that actually only one protein can fit in. Okay, and then you can check, for instance, you can use microscopy, electron microscopy. Now you can use cryo-electron microscopy to get the first, do you see one protein per nanodist? Do you see 2 proteins per nanodist? So you get already something like, is it a protein complex you are studying, or is it a single protein that you can study? Then basically with this monomering, it's just, you don't need to go into the details with this. This is a method where we use protein separation on a gel, but we don't disrupt this protein interaction with detergent. We do basically this separation under so-called native conditions. So we don't use denaturating conditions, whatever. And if you would have basically oligomers, they would be stable under those conditions. And if you have a monomer, it would be a smaller protein complex. And what you see is basically that in the nanodists, you have basically here very heavy nanodists. And under non-denaturating conditions, the original protein is actually a dimer, but it reconstitutes in the nanodist as a monomer. So you start with a purified protein that is actually a dimer, but when you reconstitute it under very strict conditions, you can reconstitute it as a monomer, and the protein is still active. So that means the protein itself, it's active as a monomer. Nevertheless, it forms oligomers in nature. Okay, so now what can you do else? I told you you can, for instance, immobilize these nanodists, and now you can add to these nanodists that contain proteins, interacting proteins. And you can observe whether these interacting proteins bind, and how fast do they bind, and how strong do they bind. Do they stay bound, or do they not stay bound? Because when you have here, for instance, a flush of buffer with the proteins, they might bind. If you then flush buffer without protein and the protein is not strongly bound, eventually they will leave again. So you would see this basically with a decrease in the signal that you detect there. And what we are using here is surface plasmon resonance. So what you use is basically light to excite electrons in a surface. And that gives a wave, an electron wave here on the surface. And this wave is very sensitive whether something is attached here, because that disturbs the wave on the surface, or not. So for physicists, this is probably very easy to understand with taking basically material, considering that you have a surface like a metal with free electrons that are basically floating in this, and you put photons on this, you can excite these electrons and they go under basically oscillation. And this oscillation is influenced whether material has been attached or not, and in this way, and how much material has been attached, and this gives signal changes. And for us, as biologists, we only look for these signal changes. As a physicist, you might even like to exactly understand what basically is the reason for these changes, and what changes you can expect. No, because as a biologist, you hope you want to get a huge signal, but maybe as a mathematician and physicist, you actually model this, and you say, Thomas, actually, if you have all your molecules there attached and a small ligand binds, that gives only a very small change. And then I would know maybe that is not a good method to use, because to understand these interactions, I want to have drastic changes. Or your calculations can come out with the result, you fairly see you should expect a strong signal. So then we would try and come back with the data, and you would reanalyze the data. And then you know that, of course, with these, like the speed, how fast the signal increase tells you something. You can calculate affinities together. Yeah? So these are, and you can basically also how stable the signal is. You can take dissociation constants again. So all of this information is extremely important, because if you now add inhibitors, from these changes, you can also say how strong is the inhibitor really preventing the interaction. Okay. Good, so the next thing what you can ask is basically, okay, what would be the structure? No? Let's assume here, this is still our pump, Tanma is our pump, and a protein needs to bind. No? Tanma could basically bind it like that, could also bind it like that. bind it? Yeah, so he looks different, no? No. And we want to know, because that tells us a little bit where's the place where it can bind. So we need structural information. Okay, one way of getting structural information is basically scattering. That's something you as physicists have completely understood. So the theory of scattering is probably the most best founded theory. We understand how basically light interacts with particles and what would be then the result of that. And that helps basically biologists to analyze the structure. But there's now here one problem, right? So if you put, for instance, here a type of source, photon source, could be X-ray, or you take neutrons, they will hit this object and they will be scattering to the side. No? But this is, of course, now not the naked protein. So it's not Tanmai that we see on. We see basically here this whole nanodisk with the belt proteins and the lipids and Tanmai. But actually we only see a part of Tanmai, because the photons would be scattered here by the lipids also and by the belt protein. And I would not see how you look in the membrane. I can only guess a little bit from these signals that I get how you would be the extracellular part. But in principle, we are in this case not interested about the lipids. We are not interested about the belt proteins. We are only interested about the protein. How would the protein in full look like? Okay, that means what we want is, we want to make the membrane proteins and the lipids invisible. And only the protein visible. Now the good news is, with scattering, you can do this, in particular with neutron scattering. I don't know how much you have discussed maybe in physics scattering. So neutrons, on what would they scatter if you take an atom? An atom, let's even go, how is an atom, if you make it very simple, don't use quantum mechanics now, make it very simple. How would you consider an atom to look like? So there is a nucleus, proton, and electrons around. Anything else than protons? Neutrons. Depends on the atom. Neutrons. Okay, so you have neutrons, protons, and electrons around. We would call this Bohr's atom model. You can consider the electrons on shells. No? And of course, it's all against physics. No? Because then you have electrons rotating. This cannot be. There should be actually magnetic field generated. They should lose energy. But somehow a miracle happens, and to understand this, then you need quantum mechanics, where the electrons are basically stable on their shells. Yeah? But for many purposes, also already on the level of chemistry, that would be fine. Okay, if you now take neutrons, on what would they be scattered? On the nucleus or on the electrons? On the nucleus. And if you take photons, X-rays, on what would they be scattered? On the electrons. On the electrons. So you see, depending on the type of light source you take, or beam source you take, you hit different properties. You either interact more with the electrons of the atoms, or you interact more with the nucleus of the atoms. Yeah? So now with neutron scattering, you can basically make parts invisible, simply due to the fact that neutrons are differently scattered on different atoms. Okay? Let's take this, you have to excuse me, because maybe you as physicists say, Thomas, this is not a good analogy, but I just try a little bit, but don't take it too serious. Yeah? What we want is basically, we want to make the lipids and the belt proteins invisible, and we only want to see the protein. And what we are going to use is so-called contrast matching. Okay, contrast matching. And what can you have actually used this even in this course. Do you remember where you have used contrast to make something visible? You don't want it there contrast matching, but you wanted to have contrast. What was the secret there? phase contrast, and what else was, maybe there was a little trick extra in preparing these GUVs and resuspending these GUVs to make this contrast even stronger. Yeah. Do you remember the two solutions? Maybe you would probably need to look back in the protocols. But I think Rajani used two different solutions. You make GUVs probably in sucrose solution, and you resuspended them in glucose. Two different solutions. So like the stick here, air and this. So you get basically, when the breaking index, in this case you use light microscopy, when the breaking index is different of these two solutions, you get contrast. You see basically on the border of these two solutions a difference. So what you have here is basically, if you consider, this is the little pipette here, and this pipette you basically put into water, you clearly see still the pipette. But if you put this pipette in another solution, which is close to the breaking index of the glass, it becomes invisible. That's contrast matching. So now you don't see anymore that there's a pipette. And this is what we want. We don't want to see lipids. We don't want to see bad proteins, but we want to see the protein. Okay? So what you do is basically, in the case of neutrons, you can take advantage of an isotope of hydrogen, and that's deuterium. You know what the difference between hydrogen and deuterium is? Yeah, what would it be? You have basically an extra neutron. No? So because chemically it's the same thing. You have one proton, one electron. But physically, you have in one case one proton, one electron, in the other case one proton and a neutron and one electron. Biologically, this behaves the same. Physically, it's different. One is heavier than the other, right? One scatters differently. When it hits, a neutron comes to hydrogen, it's to the proton. It's a different interaction than coming to a proton-neutron complex. So the scattering will differ. Okay, so what you can do is now, you basically change the biological molecule not to containing hydrogen, but you replace the hydrogen by deuterium. And you can do the same with lipids, okay? So to do this, you can either ask the chemist, make basically my proteins, make my lipids containing deuterium instead of hydrogen. Or you can basically feed bacteria with deuterium precursors, and they would start to make all these organic substances that they need. They would make a plasma membrane, lipids containing now lipids with deuterium, and containing proteins with deuterium, and then you can isolate and purify this. Yeah? So that's basically the thing. And the idea, just you see, so that would be now our belt proteins deuterated. So on many places where normally you would have hydrogen in this molecule, you have just deuterium, because it has been in the biology equivalently be incorporated in this as no differences biologically, right? You can also say isotope labeling if you want. It's isotope labeling. And you can do with the lipids, and then you get basically, you get a complex, but now composed of proteins and lipids decorated with deuterium instead of hydrogen. All the bondings, it's now between the bonding of carbons with deuterium instead of carbons with hydrogen. If you put your protein, which is not deuterated, it will basically all contain hydrogen on the places where the hydrogen should be located. Okay, now you put the whole stuff in deuterium instead of water. Then you get scattering from the deuterium. So you get scattering from the deuterium water, you get scattering from the scaffold protein, and you get scattering from the lipids. That is one type of scattering because the neutrons interact with the deuterium. The other neutrons that hit your protein, they meet now only hydrogen, the protons. And that gives a different scattering. If you now subtract the background from your signal, you only see the protein. Isn't that wonderful? So now you have a super complex protein, lipids together, but you only focus on one molecule that you are interested in. And of course you can play around. What else could we do? We deuterate our protein, but don't deuterate the lipids. Then you only see the lipids. Or you only deuterate part of the lipids, don't deuterate part of the lipids and look whether they specifically interact with your protein, form clusters or whatever. Or you don't deuterate the belt protein, then you see how the belt protein is actually wrapped around. So you can play with this, and I think we had one or two colleagues that were very much interested in scattering. So that would be neutron scattering. And X-ray scattering, you don't have this nice possibility, but you can learn from X-ray scattering, from photon scattering, also very interesting aspects, for instance, of the size of your particle, of the geometry of this particle. Okay? So that's one example, and not that you basically need to go into the details now, it's just one way you ask the chemist or you ask the biologist. So in the biologist would take cells, grow these cells in deuterated medium, add deuterated sugars, deuterated nutrients, and then you get basically all the proteins deuterated. There's a little bit basically some tricks. Just as information, this deuteration should be different between the amount of deuteration, should be different between lipids and proteins to really get a so-called matching point. So it's not enough fully to deuterate. You need to be, like on a level of 70% deuteration. The rest should be still hydrogen to get this matching point that this material becomes invisible. But these are details for those that really want to dive into this technique later. Okay, so then you can do the same with the lipids. So you give them deuterated glycerol to eat, deuterated choline to eat. Glycerol, remember, was the main molecule, the backbone molecule for the glycerolipids. This was this alcohol where the fatty acid would be esterified, the phosphate would be esterified. If you get them glycerol, the glycerol would be, in this case, deuterated. The head group would be deuterated. If you put them in deuterated water, also the lipids would start to get partially deuterated. So in this way, you get all these components that are needed to build up a nanodisk that would contain deuterated components and the non-deuterated component that you are interested in, that you want to make visible. Okay, here's just to show basically also the level of deuteration. You can basically make deuterations only in the head group. You can make deuterations in the fatty acids. You can only make deuterations in the fatty acid. That would mean I can make lipids visible at different parts. I can make the backbone visible, how do the fatty acids arrange, or I'm only interested how are the head groups arranged, how they are packed, what happens when I mix two lipids, how would this affect the head group of one. So you get really structural information on a detail, and then you have, of course, now we learned about how to simulate all of this, because in principle we know the physics behind interactions. We know what drives lipids together. We know what basically makes them moving. And all these physics law, you can make a modulation. We have seen yesterday, I think, a very good example of how you can do this. So you can model, and you can ask an experimentalist, measure, and then you compare. And also we learned yesterday, it's sometimes good to have experimental data because you need to fit some constants. That not, you have too many free constants and you need to fix some that are biologically relevant. Otherwise you would have an endless space, but biology is maybe only using a very narrow space of options, and that you can basically then narrow down by the help of experiments. Okay, cool. So that's here to illustrate, if you have these nanodisks and you have deuterated them, the bad proteins and the lipids, and the more let's say deuterated water you add, the less do they scatter so that they become nearly invisible. Okay, so this is just to give you one example. Of course, nanodisc technology you can use for many things. You can use for the protein studies. No? You can also use for drug screening if you want. If you have, for instance, a particular target protein that you want to inhibit, you can use them for structural analysis based on cryo-electron microscopy. You can also make enzymatic assays. So you can basically generate these nanodiscs with a protein that has an enzymatic activity and now can study this membrane protein in a solution like a soluble enzyme. And you can think about nanomedicine, nanodiscs as drug delivery. You would inject nanodiscs filled with a lot of hydrophobic drug that can now basically be made water soluble. Or you can think about biosensors. Okay, now in the second part, I want to, just as an example, introduce you another problem. We talked about that membrane proteins do interact with lipids and sometimes with very specific lipids. So they might even be required for the function. Then it would be a so-called cofactor, we say in biology. So Tanmai would be there, our membrane protein. He would basically be nicely folded and so, but inactive, until a particular lipid would bind. And now he would get active. Why could this be interesting in biology, or why would biology do some things? You remember that we talked about that the lipid composition of membranes differs? And you might remember that most membrane proteins, they are born in the endoplasmatic reticulum. So that means Tanmai was born in the endoplasmatic reticulum, but at a certain moment needed to be taken to the plasma membrane. Okay? Without now going what potential mechanisms could work, but you have heard one mechanism could be vesicular transport. No? So he would have a task at the plasma membrane, but he's born in the endoplasmatic reticulum. Should he be active there? No. That would be a pity, right? You should not waste energy. You should not do anything there because your task is to actually act on the plasma membrane. On the plasma membrane, you should pump protons. But you should not do this on your birthplace. So you should be a quiet baby. And then we bring you to the plasma membrane, and then we make you active. And if it would be now a lipid, what lipid could it be that you find in the plasma membrane, but not in the ER? Do you remember some steroids? Steroids? Because they were very high. You find them also in the endoplasmatic, but very low. But steroids could be, huh? So maybe only when I bring you here in this membrane, because you need steroids, you need many steroids around you, then you would become active. That could be. Or it could be glycolipids that are hardly to be found here, but in the plasma membrane. It could also still be a lipid that basically is localized in the ER, but not a very low level, and you need a threshold amount. You need not only 1, 2, but you need many amount. Yeah? So you see, in biology, this could be a trick of a place where something is born to keep it silent and only to make it active, and biology is doing this. It would be stupid that everything what is born on one place but needed in other places would already start to be active on the birthplace. So we would be interested here, for instance, now, what lipids are around such a protein? What are lipids that are really, really tightly bound? And we discussed this already, and I come in a minute to this, which trick you can do. So we talk about basically lipids that are in the bulk. Here sits Tanmai, and they are all lipids all over. We talk about lipids that are, tightly bound. So those lipids that are here around, they will exchange, and another lipid can come. But it might be that there's a specific interaction that keeps a particular subset of lipids around him that are bound lipids. And then there comes be superbound lipids. They are in a binding pocket. And if you add detergent, what do you think? So when you add detergent, of course, you disrupt all these interactions. This interaction, the higher you come with detergent, you also might disrupt. This interaction might be long-lasting, right? Because even in high concentrations, the affinity to this particular bound lipid might be very strong, and you need very harsh conditions to get it off. So if you want to basically study this, you could maybe use detergents. Carefully, but you would wash out all the bulk lipids, all the annular lipids, but not these bound lipids. If you are interested in these annular lipids, then you cannot use detergent. No? But you remember the cookie cutter, these polymers that can basically, I don't want to take my belt off today, but that can basically, cut out. So that would be, if this belt is at least large enough to accommodate the protein plus some lipids, I would have a good chance to get these annular lipids extracted. So if I basically use this type of copolymers, add them to my membrane, I would hope to catch out my membrane protein with the lipids that are close in the vicinity of this membrane protein. Okay? And that would depend, when I then analyze what lipids are there, that would tell me something. Are these just all the lipids that I can find in the membrane, or is it a subset that forms a particular shell around my membrane protein, which is important for the function? Okay, so you can basically, and you see here it's a bit weak with the actually illustration. You have again a type here of hydrophobic part and hydrophilic part, and that's important that these polymers can interact with the lipid, hydrophobic part of the lipids, and interact with the water phase. Okay, so what can you do? You take cellular membranes, you add your polymer, no detergent involved, no detergent at all, and you get your polymer discs, we call it, just polymer discs. And those polymer discs you can then basically isolate, and you can basically use them to study now your solubilized membrane proteins associated with the lipids. And when you do so, you can basically do lipid extraction. You remember how to do this? So let's say you have now isolated these type of polymer discs. You might even have purified polymer discs only containing your protein of interest by affinity. We talked a little bit about how you can use affinity. How would we get the lipids from this type of particle? It would be an organic solvent extraction. So we would basically use organic solvents where the lipids would basically then be soluble, while the membrane protein would default, aggregate, and the water would stay out of the organic phase. And then you basically can analyze those, for instance, by mass spectrometry. So then you go to the chemical department and you have a friend who says, no problem, we take the lipids into mass spectrometry. You know just very roughly how it would work. If you put a molecule into the mass spectrometry, what do we measure there actually? Or what is the machine doing? Yeah, charge is something, okay. Can very few words. If I would be your bachelor student, how would you explain to me very simple words mass spectrometry? Someone wants to try? There was something with charge, so it was good, yes. If the particle, so obviously what happens is we take advantage of the fact of an electric field again. So that's the first thing. We take advantage of an electric field, and we know that charged particles will move an electric field, but we know from physics that this movement will depend from several parameters. It will depend from the charge, it will depend from the mass. And the longer I let it run in an electric field, the more it will be basically deflected. It will change the direction, but that will depend on these parameters, and that will be basically can be measured, and I can from that conclude back on the mass. That's the way. Of course, it can be a little bit more if you have a very good chemist with a good machine. He makes some tricks. He not only lets the whole molecule fly, but he breaks this molecule into parts. No? So he lets it collide, And then you get from a lipid, so let's say what parts of a lipid could, if you break a lipid into pieces, what pieces can you expect? Let's take a glycerolipid, and you put it into pieces. You can let it fly as a whole molecule, then you get the full mass. If you start to break it down because you let collide them together, what can come free from a glycerolipid? What are the major components of a glycerolipid? Fatty acids. What else? The head group and the glycerol. So if you do it harsh, in the end you would expect fatty acids to be detected, glycerol to be detected, and the head group to be detected. And this additional information will help if you have very similar molecules still to distinguish between. No? Because only the mass, it's for instance difficult to say on which position is the fatty acid. No? It's not so easy. So you get only an overall fatty acid composition, but you cannot tell whether this particular fatty acid was on the first position or on the second position. This information isn't. But that's often also for biologists not necessary because you are more interested in the overall type of lipid. So what is the head group of this lipid? Okay, good. Yeah, then actually with this, I would also summarize. So you can use these copolymers, for example, to actually get an idea of the native lipid environment or of the complex. What proteins would be associated with your protein of interest, or whether your protein of interest would be in one copy or in several copies organized. And so very similar is basically the applications, but with the real advantage that for the core polymers, no detergent involved. Of course, then you would say, Thomas, then why have you talked about detergents at all? If there's already no copolymers, we would only need to talk about copolymers, right? If this is, obviously it's not the holy grail, we say in Europe. There's obviously also advantages and disadvantages for both techniques. So detergents are still very valid in many, situations. But copolymers can be useful, and typically what also for you maybe, it's always good to use different approaches to test the same answer, a question. That's important. So if you see typically very good papers, very good publications, they not only use one method to verify or disverify a question. Even in modeling, you have options. We have seen yesterday, you had several options to model the system. And you might have a reason to prefer one, but there could be also reasons to test the other. So if they give the same outcome, good. If not, you need to think, what is the reason and what makes, from the biological point, more sense. Yeah? So here, basically, why not using detergent plus copolymers to analyze your protein of interest? It will give you different information, but in the best, there will be some overlapping information that verifies also the two techniques. Yeah, then thanks a lot for this. And I think we have time, and I don't know how you feel. We could do the following. Because now we would normally do a tutorial, so that means you can now finish and stop. No recording necessary, and I can get rid of the microphone. So we start the tutorial session, the lecture session. And then we have a break, and you can already discuss. So one way we often do, and you probably use this technique also,